Hello, everyone, and welcome to today's live broadcast, Implement High-Throughput 3D Image Analysis for Samples from Subcellular Structures to Spheroids, presented by Oksana Sarenko, PhD, and Stephen Luke. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Molecular Devices. Molecular Devices is one of the world's leading providers of high-performance bioanalytical measurement systems, software, and consumables, and is dedicated to helping customers unravel the complexity of biological systems. Included within a broad product portfolio are platforms for high-throughput screening, genomic and cellular analysis, colony selection, and microplate detection. These innovative instruments, combined with scientific expertise, assays, and analytics tools for protein and cell biology, enable scientists to explore life's most important questions. To learn more about Molecular Devices, please visit www.moleculardevices.com. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Thank you. Before we hear from the presenters, I would like to introduce Grisha Chandy, PhD, Senior Product Manager at Molecular Devices.
Hi everybody, I'm Grisha Chandy, Product Manager of Cellular Imaging here at Molecular Devices. I'm extremely excited to have you join our webinar discussing the implementation of high-throughput 3D image analysis for spheroids as well as subcellular structures. I'm going to go over an overview of how you do high content screening, what that means, and how the MetaXpress Micro and MetaXpress software packages fit into that workflow. And then we'll turn it over to Oksana, who will be reviewing a couple of very exciting screens of spheroids done in 3D, so 3D data out of those screens. And then Steve, who will be discussing how you set up a 3D assay for subcellular structures. And then we should have some time for a Q&A. High content screening or high content imaging is an aspect of automated imaging. The process with a conventional microscope might be to put your slides on a manual microscope or a semi-automated microscope with the automated stage, or maybe some automated focusing. You acquire some images, and then what? How do you make sense of that? Those images that you collected? You need to piece together some software and analysis routines, maybe write some scripts, and all of this workflow can typically take hours. Now with high content screening, you can acquire the wide variety of assays as you saw build with that stack. You can acquire those different applications from slides, from 6-well dishes, from 3D4, from 1536-well plates. You acquire it fast at very high throughputs. You have image analysis that can happen in parallel or in the same time as acquisition. And you can turn those numeric data into meaningful results. The complete solution for imaging starts with acquiring your images. And for 3D data, you will be acquiring Z stacks of data. That data will be acquired in Z. You want to identify objects. You want to do that image analysis, which Steve Luke will be showing you some examples of. And you want to do that in 3D space, not just 2D. You want to turn those images, whether they're 2D or 3D, into multi-parametric data output. You want to get a number of measurements out of your images, and we'll be talking about how you do 3D counting, 3D segmentation, 3D volume and distance measurements. And then you want to make sense of all those numbers in this overall high content workflow, make sense of the data, either hit finding or EC50s as examples. Molecular Device provides a complete solution for that imaging workflow, starting at the top of your screen with the MetaXpress systems, which I'll talk to you a little bit about in the next slide. You have a data management solution that's automatically part of that workflow that the images go into. MetaXpress automatically talks to that data management solution, MDC store, puts the measurements that are made on those images into that same location, and then you have a way to data mine that with a QD Molecular Devices products enable hundreds of automated imaging applications. All of these assays are being done by some of our customers. We like to say we enable apoptosis to zebrafish. You've got a biological problem, we can solve that in an automated workflow for you. Angiogenesis as an example. Co-culture assays. Metagel cell culture assays. Very complicated 3D assays to be able to quantify that. Mitochondrial localization, you'll see a little bit of a mitochondrial assay in Steve stock. Stem cell assays, and zebrafish assays. So from apoptosis to zebrafish are things that people do with our products, and we try to enable those and make them easier and faster. We enable these hundreds of applications with a portfolio of intelligent imaging solutions that allows you to do breakthrough research. The MetaXpress Micro XLS is a proven product recognized in the high content imaging space. Our newest offering is the MetaXpress Micro 4, which we've just released, which takes the best features and benefits of the MetaXpress Micro and brings that to market along with the ability to be field upgradable confocal. So it allows you to grow with your needs. And the MetaXpress Micro Confocal product, which we launched at the end of last year, is a product that's ideal for some of the 3D assays we'll be speaking about. The other enabling tool is MetaXpress, which not only runs the MetaXpress systems, but is also the image analysis toolbox. You can do turnkey applications with our application modules, things like neuroid outgrowth and cell cycle, and all the way on the continuum of increasing flexibility and complexity to running journals, which are our macros, which enable powerful hardware, software decision making as part of the automated workflow. This webinar is going to focus on custom modules, which is something that bridges the gap between application modules and macros, that once you've written a custom module, it can be rerun as an application module. Talking to you about how you do 3D application modules, but in the past these custom modules have enabled things like mitotic, spindle, and centrosome detection, label-free proliferation assays, not just neuroid outgrowth, but identifying puncta on your neurites and even in migration assays. So on that note, I will turn it over to Oksana, who will be talking to you about some of the 3D assays she's done on spheroids.
Thank you, Dr. Chandy. Now I'd like to introduce Dr. Oksana Surenko, research scientist, and Stephen Luke, applications scientist, both from Molecular Devices. Oksana is working on development of new technologies for imaging and high-content analysis. She has prior experience working on drug discovery and drug development at several biotech companies in the San Francisco Bay Area. The focus of her current scientific research is on development methods for high-throughput in vitro toxicity screening, including 3D models. Oksana has a PhD in biochemistry and bachelor's degree in biology. Stephen is working as a customer representative for the high-content screening software team. He has a bachelor's degree in biology from Niagara University and more than 10 years of experience in high-content analysis and cellular imaging, starting in the field as a core imaging facility manager at Thomas Jefferson University in Philadelphia. Please join me in welcoming Oksana Surenko. I will now turn the presentation over to her.
Hello, thank you everyone for joining our webinar. I will be happy to present our studies about phenotypic characterization of compound effects on liver spheroids using confocal imaging and 3D image analysis. This work was done in collaboration with Cellular Dynamics International. Cell models becoming more complex in order to better mimic in vitro environment and provide more biologically relevant systems for drug discovery and toxicity testing. There is increasing interest in exploring three-dimensional spheroids for modeling development and tissue biology. Accordingly, development of highest throughput assay using 3D cultures is an active area of investigation. The goal of this study was to develop and evaluate high-content imaging methods to investigate toxicity effects on the morphology of liver spheroids using techniques for confocal imaging and 3D image analysis. Induced pluripotent stem cells derived hepatocytes present a valuable model that can closely resemble the phenotypes and functionality of primary hepatocytes and also minimize variability and other limitations. We've used iPSC-derived hepatocytes from Cellular Dynamics International for this study. Scientists from Cellular Dynamics, Michael Hancock and Kobe Carlson, developed a protocol for cell culture and formation spheroids. I-cell hepatocytes were preplated first in the 6-well format, then after 7 days plated into the low attachment in sphero or cornean plates. Formation of spheroids was observed in 48 hours. Then spheroids were treated with compounds known to have liver toxicity for 72 hours. After that, live spheroids were stained with calcium AM, Hurst, ethidium homodimer, or other dyes. Spheroids were imaged using molecular devices MetaXpress microconfocal instrument. Images were analyzed using 2D or 3D image analysis to define phenotypic readouts, which include morphological changes of spheroids as well as intensities and also count of different cell types. We use previously developed staining protocol and concentrations and one-step dye addition that eliminates the need for fixing cells or repeated wash steps. You can see here images of representative spheroids stained with a mixture of 3 dyes. Calcium AM was used to measure cell metabolic activity, viability, and variety of morphological parameters. Hurst was used to measure total cell count and nuclear shape, and ethidium homodimer was used to mark dead or necrotic cells. Here presented composite images of spheroids treated with compounds known to have liver toxicity. You can see phenotypic changes as a result of compound treatment. Common issues for 3D imaging described by others include efficiency of light penetration into the dense object, like spheroids, also light scattering by cells and high background from out-of-plane fluorescence. These issues tend to be less important for smaller spheroids, like used in present study. Confocal microscopy provides efficient background rejection and sharper images. In this study, images were taken using automated confocal microscope with a large field of view camera. The system effectively captures spheroid in a single image using 10x objective. At this magnification, we typically took from 11 to 17 images, 5 micron or more in distance. Images acquired with 20x objective provided even better visualization of details. In the present study, 3D analysis of images was accomplished using recently added features of MetaXpress software. The software converts a stack of 2D images obtained by confocal acquisition into 3D space with appropriate detection and segmentation of objects. Spheroids can be characterized by their size and also intensities. On the graph, you can see measurements characterizing spheroid as a single object. You can see spheroid treated with methyl mercury is different in size because of toxicity-induced loss of spheroid integrity, and also you can see decreased viability staining, like intensity for FITC channel. Individual cells and subcellular objects can be analyzed and characterized within spheroids. Custom model editor typically used for the analysis. The analysis included cell scoring for defining cells, nuclei, or cytoplasm, positive or negative for selective markers. On this slide, you can see segmentation of cytoplasm. And here you can see analysis masks. Cell scoring analysis allow to define spheroid objects in light blue, also live cells shown in yellow, dead cells shown in blue, and cytoplasm positive for calcine AM in pink. Here we present an individual Z-planes. Individual Z-planes were first segmented and analyzed as 2D images, for example for nuclei count or cell scoring features. Then the objects were connected by best match function with user-defined maximum that allowed for displacement of each object relative to another object. For example, about 5 microns for the maximum displacement for the nuclei and 20 microns for cytoplasm. Nuclei or individual cells were segmented and scored in a contents of layered 3D volume, ideally without missing objects or counting the same object twice. These steps allow defining and counting the number of total cells, number of calcine AM positive or negative cells, number of ethidium homodimer positive and negative cells. In addition, the intensities of individual cells can be defined in different colors, as well as the volumes, diameters, distances between the objects. The larger spheroid mask can be used to define number of smaller objects within spheroid or externally. Significant changes in spheroid phenotypes and cell content were seen after treatment with hepatotoxic compounds. Many spheroids lose their spherical shape, appear disintegrated, loose or flattened or irregular, have cells detached from the main body, or show condensed nuclei due to cell death. Quantitative analysis of images include finding parameters that allow assess morphological features of spheroids and also their content and complexity. The volume, diameter, and intensities were measured, and also the number of calcine AM positive cells as live, the number of ethidium homodimer cells as dead, and also average volumes and intensities of cells positive for these markers. Concentration-dependent decrease in a number of viable cells was observed as a result of compound treatment, and also increase in a number of dead cells. Average fluorescent intensity for calcine AM was dramatically reduced for entire spheroid and also for individual cells. The average volume of cytoplasm stained with calcine AM was also decreased. The number of metabolically active calcine AM positive cells, the volume of positive cells, or the fluorescent intensity provided the greatest sensitivity. Spheroid diameter, volume, or count of dead cells changed significantly but typically did not follow 4 parametric concentration-dependent responses. In contrast, the number of live cells as well as calcine AM intensity or volume of calcine AM positive cells resulted in significant assay windows and successful fit into 4 parametric dose-response curves. For the study of cytotoxicity mechanism, we also evaluated apoptosis and mitochondria integrity. Activation of apoptosis using caspase 3,7 dye was measured 24 hours after compound treatment. You can see typical activated caspase staining pattern for cells treated with methyl mercury. Mitochondria potential was evaluated with MitoTracker orange dye. You can see decrease in stain for spheroids treated with antimycin A. Spheroid assay was characterized using a representative set of about 50 compounds that included known cytotoxic and hepatotoxic agents, plus 6 chemicals considered safe or non-toxic. Table here presents selected set of hepatotoxic compounds. Known liver toxins like aflatoxins and rotenone, methylmercury, anti-cancer drugs like mitomycin, doxorubicin, antidepressants like haloperidol, and other types of drugs. We also compared effects for selected compounds on spheroids made from transformed liver cells. HepG2 spheroids were larger in size per same cell number plated, since HepG2 spheroids appear to continuously grow in a culture during compound treatment. Also, there was marked decrease in the size of HepG2 spheroid as well as decrease in a total cell number, which was in contrast to the not dividing I-cell hepatocyte spheroids. A comparison of IC50 values for compounds between two cell types is given in a table. There are significant differences between the two cell models with respect to antiproliferative compounds. The IC50s for HepG2 were in nanomolar range, whereas IC50s for hepatocyte spheroids were significantly right-shifted. In contrast, for majority of hepatotoxic compounds that don't inhibit replication, for example, ketoconazole, showed similar effects in both cell models. In a summary, 3D spheroid cell model combined with high-content 3D assay show promise as a sensitive and reproducible screening tool for assessing hepatotoxicity. And while in vivo toxicity assessment remains the gold standard, 3D models can accelerate the process by allowing rapid compound prioritization for in vivo testing and establishing mechanism of action. In addition, I would like to briefly describe another model. Similar approach was taken for imaging and analysis of spheroids in semi-solid media. Images were acquired using confocal option. These slices were captured 5 to 10 micron apart. Several Z-planes presented here show how the focus changes for different objects when moving in 3D dimension. Analysis allowed defining cells in different images and then connecting those in 3D space. Custom model allowed count and characterization of individual spheroids and also count numbers of live and dead cells within spheroids. Compound treatment resulted in decreased numbers and size of colonies and also decrease of the number of live cells and total cells. 3D analysis links the object in different Z-planes and also enables characterization and morphometric analysis of spheroids in 3D. You can see concentration-dependent effects and 4-parameter curve fits for selected compounds. The number of spheroids, number of live cells, total number of cells, number of live cells per spheroids, all those readouts have a good fit into 4-parameter curve. I would like to acknowledge scientists from Cellular Dynamics International, Michael Hancock, Kobe Carlson, and David Mann for their contribution for this project, and also scientists from Molecular Devices, especially Trisha Mitlo for technical help and others for development of instrumentation and software. If you have additional questions about cell culture protocols or staining, imaging, or analysis, please contact myself or David Mann at Molecular Devices or Cellular Dynamics. Thank you so much for your attention.
Thank you, Oksana. 3 poll questions will appear on your screen. Please select your answers, then close the poll questions by clicking on the X in the right corner. Thank you. I will now turn the presentation over to Stephen Luke.
"3D analysis can be added to different scales of samples." Oksana is talking to us today about relatively large samples, multicellular analysis in spheroids. Her presentation will also be a scientific discussion on the benefits of 3D analysis. I will cover a different set of material, concentrating on subcellular analysis, and rather than a scientific presentation, I will discuss how you might approach building a volume module. This is the set of wells acting as a backdrop for making the module. It is a simple toxicity assay with retinone in 1 to 10 dilutions and a control well. These are some of the sample images. The control treatment is on the bottom left, showing healthy, stringy mitochondria. And the 5 micromolar concentration of retinone images are on the top right, showing clumpy mitochondria. It is important to note that for some assays, you can create pretty simple modules to get a measurement to classify cells one way or another. For example, if all we needed to do was have a module which detects the toxicity of a treatment based on mitochondria, then rather than create a set of masks segmenting mitochondria, the module could just create a mask that covers all the mitochondria and measures the intensity variation under that mask. Intensity variation is a texture measurement, where clumpy mitochondria have fewer, smaller, brighter spots, and so have a higher variation than those that have long, stringy, relatively dim mitochondria. For this presentation, and in the spirit of high content, we will use a more in-depth module that can give us more values, like volume of mitochondria or the ratio of stringy to clumpy mitochondria. 3D data can mean different things depending on what is acquired. And there are different ways you can segment data based on what you want to get out of it. The first and most obvious approach is to work with objects as volumes. You can take advantage of the 3-dimensional nature of the data to better filter, process, and classify the objects. In this pattern, you take source images, which are just 2-dimensional planes acquired at various focal planes, and you analyze them together as a unit, and you use dedicated 3D processing steps to do so. The next approach is to process each plane individually as 2-dimensional data. Then, when done processing in 2D, you combine the results into volumes using various connection rules. This allows you to use any of the wide variety of 2D processing steps that are available, while still resulting in volume objects at the result side. Finally, your 3D data may just be a combination of 2D planes without any real connection between them, so you can analyze them plane by plane without introducing any 3D requirements. For example, if your sample is a tissue with z-steps much larger than the size of the objects being analyzed, or for 3D-based migration assays, where you just need to track cells moving from the top to the bottom of your membrane. This is the custom module editor. As a quick introduction to the user interface, on the left side of the window, you'll see a series of step cards, each of which is a processing step which are strung together in sequences to form the module. At the top of the window is the ribbon with buttons to add step cards to the module. The arrow points to a group of step cards that are dedicated for 3D analysis. On the right side of it is a slider that you can use to choose a Z-plane to view in the image grid. At the bottom is a film strip which shows a thumbnail for the results of each step. And on the last slide, I talked about a third approach to 3D images, where you can basically treat the data as just a bunch of 2D images. To do that, you can turn this option off. Processing images in 3D can take time based on the number of images you have. This section allows you to crop the images in X, Y, and Z, which makes creating the module faster, but still allows you to run the module on the complete image when it's time to do so. So let's look at the first part of our module, finding nuclei. To do this, we will segment entirely in 3 dimensions. To begin, let's look at the DAPI source image. DAPI can be quite textured, especially with confocal source images. So to ensure that we segment the image well, we will first use a 3D average filter to smooth out the sample. With the nuclei smooth, it is time to segment. Again, we will use a 3D tool. This one is designed to find spherical objects. It is capable of finding things a little more complex than spheroids, but generally is good for things where the centroid remains roughly the same from plane to plane. This is one of 2 step cards designed for 3D segmentation. The other, Find Fast 3D Objects, allows some rougher shapes, but Find Spherical Objects does a better job of finding bunched together things sitting on top of each other. The result of the segmentation is a cell map. A cell map is a colorful mask where each individual nuclei is represented by a color. As you navigate through Z, the objects remain the same color. Now we process the source image, then segment into processed image. This is a good point to review our source and our segmentation to make sure the segmentation looks correct. To do that, use the Compare Images step card and choose the original source image and the result of the segmentation. You can also open the images up in the 3D viewer to make sure the shapes look correct as a volume. The next thing we need to do is segment out the mitochondria, and if possible, differentiate between healthy and unhealthy mitochondria. The healthy mitochondria are long tubes, and neither of the 3D segmentation steps work well with tubes. So it is a good time to switch to the second approach to finding 3D volumes. Segment the images in 2D, then connect the 2D segments into 3D volumes. This gives us access to a lot more segmentation options. For this module, we will use angiogenesis objects. Angiogenesis is a good tool for finding tubes, which is great for the healthy mitochondria. But it also attracts nodes, which makes for a great mask detecting unhealthy, clumpy mitochondria. Before we run angiogenesis, though, we will add a pre-processing step. We want to separate and highlight relatively small structures from blur or smear. The LOG filter, or Laplacian or a Gaussian filter, is a great tool for this. With LOG used to highlight the mitochondria, it is time to run angiogenesis. Angiogenesis produces 4 masks. The first is a mask of the tubes, which we will be using for the healthy mitochondria. The second is called nodes, which is what we're going to use for clumpy mitochondria. The other 2 are a line along the center of the tubes and a dot at the center of each node, and we won't be using them for this module. Angiogenesis produces 2-dimensional masks. Our module requires volumes, so we need to connect the 2D masks into 3D volume maps. There are 3 connection rules to help with this. Do not connect objects generates a 3D volume map without actually connecting objects. Good for when the objects are smaller than the C step, but you still want to accumulate results in 3D. Connect by best match connects objects on neighboring planes in a 1-to-1 basis. It is best to use this when you know your objects have no branching or tree-like structures. For our module, we will use Connect by Touching, which connects objects on a many-to-many basis, allowing for complex structures and branching. We have the masks that we really need to make the measurements we want from the module now, but we need one more mask as the parent to enclose all the mitochondria and the nuclei for the cell. There are different strategies to do this, depending upon what you need to get out of the module. We have a good nuclei mask, so we could define a cell as those pixels in and around the nuclei, which would look like this. Or we could create a more complete mask that segments all of the important pixels, both nuclei and mitochondria, together to generate a cell mask, which would look like this. For our module, since we are using numbers generated by specific masks for the nuclei and the mitochondria, we could use the first approach without much of an issue. And we would prefer doing it this way, since it is much quicker, and this module is long enough as it is. But in the beginning of this talk, we mentioned a single mask that we could generate and measure intensity variance as a stand-in texture measurement that we could use if we didn't need volumes or other high content numbers. To get that measurement, we would need the second more accurate cell mask. And so for this presentation, I will quickly walk you through the generation of this more complete mask. To start generating the cell map, we will first combine the nuclei and the mitochondria into a single image to make it easier to find all the pixels we care about. We will then blur the image with a dilate to smudge out the details so that we can connect masks. And then we will segment the smudged cell pixels into a binary mask. How you do this is dependent a lot on the quality of your images. For this set, we can use a simple threshold, but there are other options to get the complex shape that you may need. What is important here is to get all the pixels that you care about, not necessarily to split the individual cells yet. After we have our mask, we will add a filter to get rid of the little things that come from noise or debris. In this example, we'll remove any objects which don't at some point touch the nucleus. The result will be our cell pixels, all the pixels that will be part of one cell or another. Splitting the cells is the next big hurdle. To do that, we will use Watershed Lines, for which we need to gather two intermediaries. A representation of the nuclei we can use as a marker to generate exactly one cell per nuclei, and a second to generate intensity peaks between the larger cell pixel objects. We will start with this second image. The intensity peaks will come from the distance map, an image where we will take each pixel's intensity as a measure of the distance from the edge of the object. We'll use it in the cell pixels mask to get the intensity peaks we need. Then, to generate the nuclei markers, we will add 3 steps. The nuclei volume mask has the objects we need, but isn't binary, and the nuclei which are next to each other don't have an empty pixel between them. So we will trace the edges of the nuclei and cut those edges out of the volume mask, and in the process turn the nuclei volume into binary markers. With the nuclei markers in place, we have what we need for watershed lines. Watershed lines will trace lines along peak intensities and will be sure to draw a line between every pair of neighboring marker objects. We combine that result with cell pixels and we have our 2D cell mask. Since this is a volume module, we will combine the 2D masks into 3D volume maps using connection rule. And this time, since we expect a 1-to-1 connection from plane to plane, we will use connect by best match. The final step is to define what gets measured, what numbers to generate. To do that, we switch to the Measurements tab, select our cell volume map as the object mask, and then proceed to add additional feature masks for each thing within the cell that we want to measure, namely the nuclei, stringy mitochondria, and clumpy mitochondria. Assign which images to measure and with which mask. We can select which measurements to make for each mask and image. For example, for the cell volume mask and the mitochondria source image, we can measure the intensity variation to get that one single measurement I've described a few times now. Then we can add and name additional measurements for each of the other masks until we have the set that we want. We can run the module and look at the result mask, measurements per cell, and ensure that we have the numbers and the masks that we need. Once we save the module, we can save it, and once saved, we can run it from MetaXpress or MetaXpress Power Core to generate the numbers over a plate. I hope this helps you build your own three-dimensional analysis, and thank you for your attention.
Thank you, Oksana and Stephen, for the informative presentation. Some poll questions will appear on your screen. Please select your answers, then close the poll questions by clicking on the X in the right corner. Thank you. It's time for Q&A. If you have a question you would like to ask Oksana or Stephen, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the Send button. We'll answer as many of your questions as we have time for. Dr. Chandy will now moderate the Q&A.
Hi, everybody. Thanks for joining. There are quite a few questions. I'm going to try and group some of the questions that are similar together. There are three questions around, for Oksana, around how the assay and cells were grown and done. So I'm going to read each of the three questions, and then Oksana will answer them all together. There was one question about how the cells are, whatever plate is used, and how the cells are centered in the plate. Let me just, maybe you can, oh yeah, can you image in a 96-cell plate in an automated fashion? On a low binding plate, the spheroid may not be at the same XY position. How can you get the, the spheroid centered? And why don't you answer those while I look for the other questions that were similar?
Hello, thank you so much for your questions. So I will be happy to answer. In fact, indeed, centering spheroids for imaging and automatic imaging is kind of very important point. And actually, we are able to mitigate it by using two types of plates. So one type of plate is in spheroid U-shape low attachment plates. They come either in kind of in a 96 format or 384 format. Those are from Corning. And another type is in spheroid plates. They are 96 format. They have flat bottom, but they have conical well, so that flat part is very narrow. So in both cases, because we have like large field of view camera, we able with no problem cover all spheroid in any location of the well in 10x magnification. For 20x magnification, it's also possible in automated fashion when we're using Corning plate, which kind of this U-shape allow spheroid actually to be centered in a well, so which allow to capture entire object without splitting.
Yeah, so that answers that question. So there's another question about whether you can image spheroids over time in the system.
Yes, actually, sure. We have done these studies. We actually monitor formation of spheroid and kind of acute effect of some drugs. So you can do it by using time lapse. So you
set up, like maybe 20 minutes labs, or to do overnight experiment, and you can monitor formation of spheroids in real time.
And then there's a question that I'll just answer, which is, were these, what system were these done in? These were all done in the MetaXpress micro confocal product. She sets up the assay and then puts the plate in the system. You can do that with a robot or by hand, and then the system is run, and you can do them either as an endpoint assay, like presented this presentation, or as she mentioned, as a time lapse. Can you discuss, a different question is, can you discuss a little bit about your staining procedure and how you added the dyes and whether you removed them and things like that?
Mm-hmm. This is another kind of important topic, because those spheroids are kind of not attached to the bottom. There are some challenges for staining and washing. So the protocol which we develop actually include just single-step addition of dyes. So usually we add like 3x concentrated dye mix in PBS directly to the wells without removal media or anything, and we allow like at least 2 hours for staining. We usually keep during these 2 hours plates back to incubator, and with those dyes which we use, it's kind of sufficient to penetrate inside spheroids. And we don't wash dye out, so we just image it directly. So this confocal system, it has very nice background rejection, so it's not a problem at all.
I remember, there's another question on the time lapse. I remember that one of the data you published was on just doing cells and sphere formation over time without a stain. Have you also done it with stains? Have you done the live cell measurement with stains?
We have not done it, but it is possible, yes. So it's just kind of the matter of to choose the right dye so it wouldn't fade away or it wouldn't cause toxicity. I would imagine that this kind of whole cell stain dyes could be useful for this purpose, or maybe if you use Hurst stain in a lower concentration, it's also kind of a okay option.
Question for Steve. These are 2 related questions. Are there tools available to estimate cell or sphere location or distribution in 3D analysis, distribution from cells or other spheroids? And then there's the direct question is, can you perform distance measurements in 3D? So those 2 questions I think are a little bit related. Steve, can you take that?
Sure. Yep. So the custom module does let you measure positional information, X, Y, and Z position. There is a measurement for each object that is the distance to its nearest neighbor. So that gives you dispersion measurements. And then there's also a measurement that you can make on, there's a 3D distance map. So you can have distance of one object from some common source or from something in a different channel or a different type of object.
Here's a question that I'll take. Can the software analyze images from other images other than the MetaXpress? Yes, we have ways to import data from third-party imagers. We haven't, there's a limited set of instruments we've tested it on, so it really depends on the format of the data, whether we can import it or not. And we've done it with some, both competitor and collaborator instruments. So it really depends on what you have. Sorry, let me go back and look at the questions again. Steve, a question for you on how long 3D analysis in a 384-well plate might take.
Okay, so that depends a lot on the module. There's simple modules like we were talking about, that single mask that you could use to measure the variation under. that can take just a few minutes, 3 to 5 minutes per 3 to 4 well plate. And then something that's a little bit more robust like that to whole mitochondrial volume, if you have lots of Z-planes, that could take upwards of, not 3 minutes per plate, 3 hours per plate. The big one could take 30 hours. It's a big investment of time. Yeah. There's also MetaXpress Power Core, which can then speed up the analysis time by distributing the analysis around to different computers. And you're looking at maybe reducing the 3-hour quick module down to 45 minutes and taking that 30-hour down to maybe 5 hours.
Yeah, you can maybe get sometimes maybe a 10x improvement, so maybe 3 or 4 hours.
Right.
There's a question. I'm just go back to Oksana. Did you use air or water or oil objectives for your assays?
Actually, it was all air objective. Yeah. No oil in this assay.
Steve, what are the 3 connection rules again?
Okay. The 3 connection rules. There's a do not connect objects. This one creates a volume map, but does not connect objects from one plane to another. So it's very good for when you have, like, let's say, nuclei in your spheroid. The nuclei might not actually appear on multiple Z-planes, but you still want to count the number through Z. And so this would allow you to accumulate data through 3D without actually connecting anything. The second rule is connect by touching. So if there's any overlapping pixels from one plane to the next, they're going to be connected as a single object in 3 dimensions. And then the third one is connect by best match. And this is going to be a one-to-one connection from one plane to the next. And it's going to use size and position to determine what object to connect. If there's more than one that might possibly connect, it'll determine the best match based on size and position.
Steve, there's a question on whether you need a confocal system, or actually it sounds like a customer has our existing MetaXpress Micro XL. Can you do these analyses in both wide field or confocal mode?
You can do them in wide field mode. You don't need the confocal. The confocal will give you improved segmentation because it's removing a lot of the out of focus light. And then the depth or the 3D size of the object might be extended, so it might be measured as deeper and the volume overestimated in a wide field system. But it's still possible to segment in both wide field and confocal.
Okay, thanks, Steve. There's a couple of questions about if this webinar is going to be available. I think they've been answered privately, but it will be available, and we'll let the attendees know when it is posted. And it's usually within less than 2 weeks. You should be notified about its availability. Actually, there's a question I'm going to have Oksana answer it. Have you done much immunostaining of the spheroids?
So I've tried staining of 6 spheroids, but with phalloidine only. I didn't try to stain for some other markers yet. It should be possible, but it may be some issues with antibody penetration, but we have not addressed it yet. It's in the future.
I'm going to take this question. The question is the 20th the highest magnification that this process can handle? What's the maximum mag at which images can be processed? There's no limit on magnification for images to be processed. I think for Oksana's assays, there was a benefit of the plate she was using and the spheroid size and things like that to do it at 10 or 20x, but the software doesn't care about magnification. I'm not sure, Steve, were the mitochondrial assays taken at 20 or 40x?
I believe these ones were 60x.
60x, okay.
Yeah.
It really depends on the assay and what you're trying to see, what mag and what objective you would use. It just happens that Oksana's were done at lower mag. So Oksana, did you do any normalization of your calcium live cell signal to number of nuclei per spheroid or anything like that?
In fact, it was done automatically because the numbers were I choose to be reported were average intensities of cell cytoplasm. So I can either get average value per well, or I can get individual intensities of individual cells. It is also possible to get other intensity measurements like integrated intensity or intensity of entire spheroid as an object.
Oksana, did you compare the behavior of compounds in 2D or 3D, and what were the results?
Yes, we have done this study. So set of compound was compared for hepatocytes in 3D system and also compared with our previous data done in 2D system with the same cells and a similar protocol. So cells were treated for 72 hours and then stained for viability markers and then analyzed by using cell scoring for live or dead cells. So with this similar approaches, we were able to get IC50s for number of compounds, and for number of compounds they were comparable, but for the number of other compounds it was different. So one example was paclitaxel or haloperidol. We get much smaller IC50s for 3D system.
Another question for Oksana. Have you tried spheroids from other cell types or organs other than the iPSC-derived hepatocytes and the HepG2?
Actually, we've published last year a paper on using spheroid assay for testing anti-cancer drug using HCT116, so this colon carcinoma cells, and it was actually working very robustly. Also, we were using different cell types from induced pluripotent stem cells. So we have done studies with cardiomyocytes, and it was actually really fascinating assay because we were able to record beating of those cardiomyocyte spheroids in a culture and monitor that.
Which readouts from the 3D analysis are best for determining IC50s? What had the best sort of Z primes and assay windows?
So we have done some studies on the topic. So if for the present study where these hepatocytes were not proliferating, the best readout was the number of live cells per spheroid, and also other good readouts which had a very large assay window and low variability were cytoplasm volume, either average per cell or total per spheroid, as well as average cell intensity. Those were very good markers for IC50s. Some other readouts, for example, size of spheroid or diameter or intensity of entire spheroid or number of dead cells was having significant assay window, but much smaller than that other which I described, and IC50 curve fit was very poor.
Did you already answer this question? Apologies if I asked already. How was compound treatment performed for 72 exposure, a daily dose, changing fresh compound every 24 hours?
Yes, actually for 72 assay, we were kind of we didn't re-add compounds during the assay, but it is possible to do if you kind of choose so. In our previous studies where we did 5-day incubation for these cancer spheroids, so we added compounds once during this incubation, like on a day 3.
Okay, we're at the hour. I know I did not get to all the questions. Apologies. So if you want to contact us directly, please do so. Our email address is our firstname.lastname@moldev.com. You can learn more about our product at our website, www.moleculardevices.com. We'll let the attendees and registrants know about this webinar directly by email. But for those who don't get an email, we will usually host something on either our website or on LabRoot's website for some time so that you can review this webinar. And we also have another place to stay in touch we have a software forum, metamorph.moleculardevices.com, where we have both modules and questions that can be answered about both Metamorph and MetaXpress, and it's an open forum and it's peer-to-peer. So that's another resource for you to learn more about our product. I will just take one more quick look at the Q&A to see if there's no other key questions, and I think it's slowed down. So again, thank you for your attendance, and I'll turn it over to back to LabRoots and Judy.
Thank you. I would like to once again thank Oksana Starenko and Stephen Luke for their presentations. Do you have any final comments?
Yes, I really appreciate for all the people who join, and I try to answer many questions, but please don't hesitate to email me additional questions you have. So we would like to further develop these techniques. And if you have questions about cells, so please contact Cellular Dynamics. And if you have a question about software, Steve Luke would be the best person to answer.
Steve, did you have any closing thoughts?
I would also like to thank everybody for their attendance. And yeah, feel free to contact me if you have questions. Thank you.
Thank you both again. I would also like to thank our sponsor, Molecular Devices, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through November 24th, 2016. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Bye.